Finally, we discuss analysis of extracted DNA. Just like your proteins, we can also perform electrophoresis on your DNA samples. This time, instead of polyacrylamide gels, we use agarose gels. Agarose gels make larger pores than your polyacrylamide gels because relatively your DNA molecules are larger and heavier than protein molecules. For further exploration, you can go to this website, the Lab Exchange website, where you can experience virtually how to perform agarose gel electrophoresis. If you remember, in SDS page, we use reference proteins or what we call protein ladder with known molecular weights to estimate the weight of your protein sample. In agarose gel electrophoresis or AGE, we also use what we call DNA ladders. But instead of molecular weights, we use base pairs or number of base pairs. So your DNA ladders would come with, for example, 100 base pair, 300 base pair, 1000 base pairs, 3000 base pairs, and so on. We also use spectrophotometry, number one, to estimate the concentration of our DNA sample, and number two, to check the purity of our DNA sample. When we discussed protein purity, you encountered the A280 to A260 ratio. And you know that your DNA or your nucleic acids in general have absorbance at 260 nanometers. That is why we get the A280 to A260 ratio. So in the case of DNA, we get the A260 to A280 ratio. So an acceptable value for this ratio would be from 1.7 to 2.0. In addition to this, we can also account for the turbidity of the solution. So sometimes when we perform DNA extraction, the final solution, final DNA solution, would be a bit turbid. So to account for this, we subtract the absorbance at 320 nanometers from the absorbance at 260 nanometers. This will give us a more accurate estimate of the concentration of the DNA in our solution. Here is a formula showing how to compute for the concentration of your DNA molecules in solution. So you have this A260 minus A320 multiplied by a dilution factor if you have, times 50 micrograms per ml. This 50 micrograms per ml is from experimental observation that one absorbance unit is approximately equivalent to 50 micrograms of DNA per ml of solution. So you can use this formula to get an approximate concentration of your DNA sample. In addition to this, some methods require organic solvents or chaotropic salts, such as your guanidinium chloride. Most of these solvents and salts also display absorbance at 230 nanometers. So it's also wise to check the absorbance at this wavelength to check for the presence of organic solvents or chaotropic salts in your DNA sample. If the absorbance is considerably high, you might have to exchange the buffer again to decrease, if not to remove, all of the organic solvent and salts in your DNA solution. Because of this, it is highly recommended to get an absorbance spectrum from 230 nanometers to 320 nanometers, instead of measuring the absorbance values at the specific wavelengths. Just like your proteins, you can also perform hydrolysis on your extracted DNA. The general mechanism of hydrolysis in acid would be first, the removal of the nitrogenous base, followed by the hydrolysis of one of your phosphodiester linkages, eventually separating one DNA strand into two strands. Remember, DNA is not susceptible to base hydrolysis or hydrolysis in the presence of bases because the mechanism involves a hydroxyl group in the 2' carbon of the sugar. Since this hydroxyl group is absent in DNA, hydrolysis in the presence of base cannot take place. Of course, we can also perform qualitative analysis of the extracted DNA and also the hydrolyzed DNA. We have the Schiff's test. This test is specific for aldehydes. So, which of the components of your nucleotide can assume an aldehyde conformation? It's your sugar, the ribose, or the deoxyribose. So it is important that the DNA undergoes hydrolysis first before it can test positive in the Schiff's test. So your Schiff's reagent is made up of the fuchsin dye. In the presence of acid, bisulfite bonds to the central carbon of the fuchsin dye. The effect of this is that this dye gets decolorized. So this dye initially has a color, but in the presence of bisulfite and acid, the color disappears. And if we add the aldehyde coming from our hydrolyzed DNA, it forms a pink colored complex. So the positive result is the solution turning into pink. Next, we have the test for deoxyribose or the dish diphenylamine test. This is specific for the deoxyribose. It will not work for ribose. Therefore, we can use this test to distinguish between DNA and RNA. Just like your Schiff's test, your DNA must first be hydrolyzed so that you have a free deoxyribose molecule. In the presence of acid and with heating, the diphenylamine reagent forms a colored complex with the deoxyribose molecule, and the positive result is a blue color in the solution. We also have test for purines. The test for purines needs ammonium hydroxide. So the reagent is, or the solution is basic, and we have silver nitrate. In the presence of a basic reagent, your silver nitrate can interact with the lone pair of electrons in the nitrogens in your purines. So if you go back to the structure of your purines, you will find that some of the nitrogens in the cyclic chains possess lone pair of electrons. So the silver ions can interact with those lone pairs. And the effect is that a precipitate will be formed. The color of this precipitate is white. Your DNA does not need to be hydrolyzed because the lone pairs found in your purines are not involved in the glycosidic bond between your purine and the sugar. So, the test for purines can detect or can give positive results both for an intact DNA and hydrolyzed DNA. We also have test for phosphates. So the test for phosphates begins with incineration of your sample. The incineration is important to remove other elements, carbon, hydrogen, nitrogen, and oxygen in our DNA. So incineration will allow these elements to be released as gases like carbon dioxide, water vapor, oxygen. Meanwhile, the phosphate will be left behind. After incineration, we add ammonium molybdate. Ammonium molybdate will form a yellow colored complex with your phosphates. So the positive result is a yellow precipitate.